Hello everyone, welcome to this video. In this video we will be talking about the 8th lesson in your grade 10 science syllabus, characteristics of organisms. In this video we will be going through all the important parts of this lesson, and this will be a short version where you need to know all the important things, and you can also use the content you see on the screen as a short note. Now without any delay, let us start off with the lesson. First and foremost, in this lesson we must understand that all living organisms have certain characteristics: cellular organization, nutrition, respiration, irritability and coordination, excretion, movement, reproduction, growth and development. So these are the 8 common characteristics of all living organisms. And in this lesson we will be discussing about each and every single one of these in a bit detailed manner. So first and foremost we are going to talk about cellular organization. Now organisms are composed of one or many cells. Unicellular organisms have one cell, and here you can see some examples for unicellular organisms: Clabinomonas, Euglena, Amoeba, Paramecium. They are made up of one cell, and therefore they are called unicellular organisms. Uni means one, cellular stands for cell, so unicellular means one-celled organisms. Multicellular organisms are organisms composed of many cells. Multi means many, cellular refers to cell, many cells. So all other organisms like humans, cats, dogs, rats, bats, they all are multicellular organisms. Now the origin of multicellular organisms is from a single cell. Now when you take a look at this image over here, a baby is formed from a single cell. The zygote is produced when an egg and a sperm fuses together and undergoes fertilization and forms the embryo. Then the embryo will undergo cellular division, and then it will increase the number of cells, forming a multicellular organism. So the origin of multicellular organisms are from a single cell. Now the next important thing is the organization of different cells in a plant body. Now this is a picture you get in your textbook, and they could question you all for your exams about it. It's very simple, so people may ignore it, and some people may not focus a lot on that. The most important part in this picture is this one over here, the part where they show the cross-sectional area of a plant leaf. So to start off, let us go through this picture. Here you can see the flower, and in the flower you will see the pollen grain, and then you get the stem, and you get the roots, and then this stem you will get the phloem cell in the stem, and you will also get the xylem cell inside the stem. And this leaf, you can see something like this when you take a cross-sectional area and observe it under a microscope. You will get the epidermal cell, the palisade you will get the epidermal cell, the palisade cells, the stomatopore, and of course the guard cells, which is on either side of the stomatopore. And here is the stomata opening. These are the guard cells on either side of the stomatopore and the stomata opening. Now this is a bit unclear, so here you can see a clear image. The parts above the ground is known as the shoot system, and below the ground it's called as the root system. You get the taproot, which is the big root, and the lateral roots, which are the small roots. Then here you also get the auxiliary bud at the tip of the branches, and you get the terminal bud or the apical bud at the very top of the plant or in the edges of the branches. Then you get the parts of a leaf over here, the leaf blades, the petiole, and then you get the stem and the flower, the pollen, and it's basically a clearer image of this. Now the next important thing is the organization of different cells in a human body. Now you can see, you just must remember the shapes of these cells and where you get them. For instance, nerve cells, they have this sort of a shape. Bone cells, they have a shape that looks like this, like centipedes. Nerve cells, they have something that looks like this, like some amoeba or some sort of a weird substance. Then skin cells, they look like this, like a collection of marshmallows, like a marshmallow pudding when you get it, or something like this, like a backbone sort of. Then gametes, an egg and a sperm. And then you get white blood cells and red blood cells and muscle cells. So that way we get many of them. We must remember the shape and what's its name. So multicellular organism possesses a tissue and a system level organization. The smallest structural and functional unit of a living condition is the cell. A group of cells modified to perform a specific function is known as a tissue. Now this is very important. We are going to learn about the different organizational levels of an organism. So we are going to start off with a cell, which is the smallest structural and functional unit of all living conditions. And a collection of cells, in simple term, a collection of cells modified to perform a specific function is what we call as a tissue. So examples for animal tissues, you get blood, muscles, epithelial, bones, nerves. Plant tissues, you get xylem and phloem. And a collection of tissues will form an organ. The tongue, heart, eye, brain, liver, a collection of tissues. A collection of organs will form a system. In humans, we have the blood circulatory system, the excretory system, the digestive system, the reproductive system. We have so many systems. And plants mainly compose of two main systems, the root system and the shoot system. A collection of systems is going to form an organism. And here we can see an example for organizational levels that can be identified in a living being. Cell, tissue, organ, system and organism. Collection of cells make a tissue. Collection of tissues make an organ. Collection of organ makes a system. Collection of systems make an organism. So an example for each, you can take heart muscle cells. A collection of heart muscle cells will form the heart muscle tissue. And then a collection of heart muscle tissues will form the heart. And a collection of the heart will form the blood circulatory system. This blood circulatory system and all the other systems will altogether form the organism. So this is an example for organizational levels that can be identified in a living being. You can take this, how the blood circulatory system is formed. You can use another example and get how the reproductive system, or otherwise the respiratory system, anything, but this is a very easy example to remember. Next up, we are going to talk about the second characteristic, that is nutrition. So nutrition is the process by which energy and materials are obtained for the maintenance of life. That is what we call nutrition. So the equation of nutrition, you can see it over here. This is something very important which you all should remember very thoroughly. So carbon dioxide plus water in the presence of sunlight and chlorophyll will give glucose plus oxygen. And this glucose is what we consider to be nutrition, the process by which energy and materials are obtained. So energy is obtained by using glucose. When glucose enters our cell, then cellular respiration will take place and then ATP will be produced, which we will be talking in the when we go further in this lesson. So this is your word equation, and then below over here you can see the chemical equation with the symbols. CO2 plus 6H2O, sunlight and chlorophyll, arrow, and you write sunlight and chlorophyll on either side of the arrow, C6H12O6, 6O2. So these two are very important to remember. So organisms that produce their own food, own nutrition by themselves, we call those organisms as autotrophic organisms. So if the organism produce the food they need by themselves for themselves, you call them autotrophic organisms. Now this autotrophism is divided into two categories. They are photoautotrophic and chemoautotrophic. So photoautotrophic organisms are the organisms that uses solar energy. They use the energy obtained from the sun to produce their own food. Green plants, cyanobacteria are examples for photoautotrophic organisms. Chemoautotrophic organisms uses chemical energy. They use various chemicals and they produce their own food. Normally some bacteria, they are chemoautotrophic. Now if the animal utilize food produced by these autotrophic organisms, for an instance plants, or organic compounds with other living organisms, we call them heterotrophic organisms. We humans, we eat plants, we eat different other organisms, so we are heterotrophic. We don't produce our own food by ourselves, but we simply consume we consume different plants and organic compounds. And now for an example you can take here. So sunlight is touching the grass produces its own food by itself and hence it's autotrophic. Grasshopper eats the grass, so it's heterotrophic because it's depending on the autotrophic organism for its food, for its energy. And then this heterotrophic organism is eaten by the frog is eaten by the eagle, then the frog can also be eaten by the snake, and then like that the food web will keep on continuing. So you all will be learning about food webs later on as well. Just remember for the moment that there is something like this. Next up we are going to talk about the third characteristic of living organisms, that is respiration. So the process by which stored food, that is normally glucose, is transformed into energy inside the cell is what we call as cellular respiration. So this equation is simply the reverse of the equation for photosynthesis. Photosynthesis, the equation was CO2 plus H2O. In the arrow on either side you write sunlight and chlorophyll, and then you get C6H12O6 and 6O2. Now here it's the opposite. First you write C6H12O6 plus 6O2, arrowhead, you can write cellular respiration, you get 6CO2 plus 6H2O plus ATP. So it's the process C6H12O6, it is glucose and it is stored in your cells. Now this stored glucose or food is transformed into ATP, which is an energy inside the cell, and this transformation of stored food or glucose into ATP or energy is what we call as respiration. During inspiration we take in oxygen for respiration and remove carbon dioxide, water vapor. Yeah, so when we inhale or during inspiration we take in oxygen, and then during exhalation or expiration we breathe out carbon dioxide and water vapor. Now we are going to do this small activity which involves a frog in it. So in this activity we are going to show the release of carbon dioxide during respiration. So first and foremost we are going to take some beakers like this. You can fix a cork on each one, you can connect some delivery tubes like this, you can see, and then here there is a suction, so you have to arrange the apparatus like this. And here into the first beaker we are going to place KOH, second beaker we are going to place lime water, and the third beaker we are going to place our frog, and in the fourth one we are going to place some lime water, last one we are going to place some water, and then we also have a suction over here. So when you do this, when you arrange this setup, you will be able to observe something in this setup. That's the purpose of a practical, to get some observation and to conclude something, right? So here you will be able to observe that B has no color change. The lime water is still colorless, but then D turns into a milky color. That is our observation. The observation is what we can detect by our senses, by seeing, hearing, tasting, smelling, those. Then we come to a conclusion. Conclusion is like what we confirm. Okay, this happened, so we can confirm this is there, or this is the reason for it. So here the conclusion is that the frog produced carbon dioxide during respiration. So remember, this is very important even for the A levels. Carbon dioxide, when sent through lime water, turns the lime water milky. That is an experiment to identify carbon dioxide. Now you may ask, that is basically it. That's all for the practical. Now you may ask, why did we use KOH here? Now KOH is used to absorb carbon dioxide. Now here there is a suction, and the suction is used to maintain a continuous flow. So first the air comes from out into the KOH, then all the carbon dioxide is absorbed, and then we send it through this other delivery tube, and what do we send? We send oxygen, pure oxygen air sort of. It's not completely pure, but most of it is oxygen, and then it keeps on going like this. And here we use lime water to confirm that this air that's sent from the KOH to lime water has no carbon dioxide. If this thing turns milky, that means the KOH hasn't absorbed the carbon dioxide. But since this is colorless, that means all of the carbon dioxide has been absorbed by potassium hydroxide. That we must understand. Now this same activity we can reconstruct it and do it by using germinating seeds. So we are going to simply take something like this. You can see a glass funnel, you can take a beaker that's like this, and then you can place some delivery tubes, take some colored water, you can insert some germinating seeds into the tubes, and these are flasks. Okay, you call them flasks. So, and then you get this small test tube or small tube where you have put a bit of KOH and colored water. Now when you set this whole thing up, you will be able to observe that colored water rises through the glass tube of A, and it does not rise in B. Here you can see no rising of colored water. Here it has risen. So we can conclude that in apparatus A, carbon dioxide is released during respiration of seeds, dissolved in this KOH, and O2 is absorbed by the seeds for respiration. So it needs oxygen, and all the carbon dioxide released has been absorbed over here. So when it's taking in oxygen, this will little by little rise. Now here there is no change because the absorbed volume of O2 is equal to the volume of CO2 released. Here CO2 is absorbed by the KOH, and O2 is also again absorbed by the plants. So there is a certain pressure difference, hence this water will rise. But here there is no such thing because CO2 absorbed is equal to the CO2 released is equal to the O2 that's absorbed. So that's what happens here. Now in this we have to assume that the volume of carbon dioxide in flask is negligible, and in respiration absorbed volume of oxygen is equal to the released volume of carbon dioxide. And another important thing is, why did we use colored water? In order to get clear observation and readings. Okay, so that's basically it for that activity. Next up we are going to talk about irritability and coordination. So the ability to respond to stimuli receives from the internal or an external environment is what we call as irritability. Stimulus is a change which is strong enough to bring about a response. And a receptor. The stimuli are detected by organs such as eye, ear, nose, tongue, skin. They are what we call as the receptors. Response is how we react to the changes in the environment, and the effectors are the responses. What are the organs or things that are used to give about our response. So that we call effectors, and there are two types, muscles and glands. Now for an instance, let's say while you're walking, your leg is being pricked by a thorn, you're stepping on a thorn, or you're stepping on some charcoal that is very hot. So in that case, suddenly when that heat, or when the thorn, let's say stepping on a thorn, when the thorn pricks into your leg, you will immediately, you might at once scream, you might jump up, you might take your leg away, so you might act, right? So that is what we call as a stimulus, a change strong enough to bring about a response. And what detected that feeling of the thorn being pricked? It was the skin, right? The skin in your foot. Then the response is how you reacted. You may have jumped, you may have moved your leg away, you may have cried, you might have shouted. That is your response. The effector is how the response is done, or responses are done using the effectors. So what was used in order to bring about such a response? Maybe the muscles. Your muscles may have made your legs move behind, or your glands may have secreted some hormones and stuff to increase your certain, or it may be some hormones or glands, or it can be something. So it may be your muscles that used to raise it up. And the communication between different organs during the response to a stimulus is what we call as coordination. So it's like the foot, when immediately you prick a thorn, the skin is going to detect it, and then it's going to send a message to your brain saying, this one stepped on the thorn. Then the brain is sending a message to the foot and the muscles and the glands telling, immediately move it, or immediately move it. So that is coordination, communication between different organs during a response to stimulus. Nerve, muscles and hormones are important in coordination. Both animals and plants respond to stimulus. Excretion is the removal of byproducts from the body that are produced during metabolism. That is what we call as excretion. Now, there are different excretory products. Sweat, which has water and salt, exhaled gases, which has carbon dioxide and water, and urine, which comprises of urea, water and salt, are examples for that. Now the main nitrogenous excretion of a human takes place in kidneys, and you might have, as we had explained earlier also, carbon dioxide is removed during respiration and oxygen during photosynthesis in plants, right? So during respiration, plants releases carbon dioxide or removes carbon dioxide. During photosynthesis, it removes oxygen. So these things also are excretory products, which is very important, and many tend to forget it sometimes. Now our next one is the movement. Movement is basically this. Organisms fulfill their requirements with the help of movement. So the method used by organisms to, you can say, fulfill their requirements, and by requirements, what do we mean? Food, protection, reproduction. Those are their requirements. So unicellular organisms use different appendages to locomote. For an instance, you can take paramecium, they use cilia. You can take amoeba, they use pseudopods. Over here you can see pseudopods, that means fake limbs, sort of fake legs. And then this euglena and chlamydomonas. Over here you can see a picture of euglena. All these three organisms use this appendage called a flagella in order for locomotion. Also that's called as movement. Next up, we must also understand that plants also show these particular movements. Now just think, in these photos, like you can see, the tip of the shoot, okay, the tip of the shoot. I explained what the shoot is in the beginning of the video. So the tip of the shoot, that means the system above ground level in a plant, will start moving towards the light. As you can see over here also, it moves towards the light. Here, towards the light. If it moves towards the light, we call that movement positive phototrophism. An easy way to remember this that I personally think is, photosynthesis, you need light for photosynthesis, right? So the plant is reaching out to the light, it's going to the light, so it's something like positive. So going to the light means it can obtain light in order to perform photosynthesis, which is something positive. It's going towards the light, so it's something like a positive thing, so it's positive phototrophism. Root apex moves towards gravity, so it's going towards the, you can say, geography, it's going towards the ground, G, ground, geo, GG, something like that. Just find an easy way to remember this, or you can sort of make up your own theories, or like, not theories in the sense like own theories on how to remember this, or you can memorize this. Just remember that plants also show movements. If the shoot apex moves towards the light, we call it positive phototrophism. If the root apex moves towards the ground or towards gravity, we call it positive geotrophism, which is basically negative phototrophism. And then we have the other one where the pollen grain on stigma moves towards the ovary. So there is a complex process over there. When the pollen grain comes onto the stigma, it forms this pollen tube. After secreting various substances, then it's transported into the ovary. So there's an entire theory about that, which you don't need to know now. And that is what we call as chemoautotrophism, because chemicals are normally involved in this. Chemoautotrophism. We call it chemoatrophism because certain chemicals are involved in this. Next up, we are talking about the seventh one, which is reproduction. That means producing a new generation by unicellular or multicellular organisms from the current. Next up, we are talking about reproduction, which is the production of a new generation by a unicellular or a multicellular organism for the continuation of their species. Now, reproduction is of two types, sexual and asexual. Sexual reproduction involves gametes, and that is when two gametes of individuals of the same species join together, undergoes fertilization and forms a zygote. For instance, you can see over here the sperms, okay, it's joining with this egg, and then one sperm will enter the egg and it will fertilize, and then a zygote will be formed. In the same way, there is a process which I explained earlier also in plants. So that is sexual reproduction, where two gametes are involved. Second one is asexual reproduction, where a single organism can produce an identical new offspring without the contribution of another organism. So it's like you can see one of these organisms. Then it will basically cut itself into two. It will divide itself into two, producing two new organisms. That is what you call asexual reproduction. Now, in asexual reproduction, something significant or important is that the dot organisms are identical to mother organisms. So here, this is the mother organism and this is the dot organism. So they are identical to the mother. So if you take, even there are worms like this, if you cut the worm into two, it will form two offsprings. Okay, so that is asexual reproduction. Now we are coming to the end of this lesson, where as we talk about growth and development. So this is very important. The increase in the number of cells by cell division, we'll be talking about cell division, which includes mitosis and meiosis, in another video in the near future. Do not worry about that. So just remember, cell division occurs and the number of cells will increase, and this contributes towards the growth of multicellular organisms. The growth of unicellular organisms is considered as the increase in size and volume of the cell. So amoeba, bacteria, euglena, paramecium, they are unicellular organisms, and their growth means the increase in the size and the volume of the cell. And in multicellular organisms, growth is the increase in the number of cells. Okay. So in multicellular organisms, for instance, one cell, and then multicellular organisms, let's say two cells value, multicellular, that organism keeps increasing the number of cells, say 4 cells. Then this is divided into 8 cells. So increasing in the number of cells is what you call as growth and development in multicellular organisms. Unicellular, like paramecium, bacteria and stuff, their growth means this volume, inside volume and size is increased. So their growth means when the volume and the size increases. Cell growth means, okay, that we must remember. So growth is what we defined over here. Growth of multicellular organisms and the growth of unicellular organisms. Then we talk about another important point, which is cell growth. Cell growth is the irreversible increase in the dry mass of a cell. Okay, so that you get a cell like this and it has the dry mass. So this dry mass will keep on increasing and you cannot reverse it. You can't like reduce it. You cannot reverse it. So this irreversible increase in the dry mass of the cell is what we call growth and development. Development means the increase in the complexity of a cell. Okay, now so long we were telling growth and development. So we talked about different types of growth, and now we talked about finally development. Okay, so growth and development can be expressed in 3 steps. Irreversible increase in the size of a cell, increase in the number of cells by cell division, and cell differentiation. So just remember them. It's not very important to know what it is and all those things. Now we're going to do activity 4 in your textbook, where we are going to observe a plant growth using an oxynometer. So you have to take a potted plant, connect it to a pulley using a string and give it a slight weight, a very mild weight, and then we use this oxynometer. Now remember this word, this oxynometer is something very important, and normally it is something they test. They might as name an instrument which is used to identify the growth of a plant, oxynometer. So it has a scale. You will get this in a lab and you have to do this practically. So you will notice, like you have to keep it for some time after fixing it like this, that the plant grows very slowly, but this indicator of the oxynometer shows it at a larger or greater scale. Now before we end the lesson, we are going to talk about something really interesting and which is very popular during these days. We are going to talk about viruses. So as you all know, viruses are very small and they can only be observed using an electron microscope, and they are about 1/1000 the size of a bacteria. So they're so small, they're even super small, so small in the sense they're so much smaller than a bacteria. 1/1000, as you can see over here. They show living or non-living features according to their status. So most of the features these viruses show are non-living features. They don't respire, they don't excrete, they don't have any sensitivity, they don't show movement, they don't have enzymes, there is no cell structure or cell organelles, it has no metabolic reactions, it has no organized nucleus and stuff. It's basically a protein layer. It's just having a cell membrane basically, and DNA, RNA inside it. And the only 2 living features you get in these viruses is that it can reproduce in a living cell. Okay, you need a live host for the virus to reproduce. So if the virus is kept outside of a living cell, it will shrivel away or it might be destroyed, but it basically cannot reproduce. But inside a living cell, say it entered a human, an animal, a dog, a cat, a rat, any animal, it can reproduce, and then it can cause diseases or mutations and various things to the human or that organism. And normally these viruses have DNA or RNA. Normally all other organisms, humans, animals like bats, cows, dogs, almost all the organisms have both DNA and RNA. But here viruses only have one of them. Now viruses infect animals, plants and bacteria itself. there are different diseases caused by viruses. Now it's very important to remember some of these things, and a very common one is COVID. Then we also have chickenpox, you can see in this picture, where you get these pink spots all over your body. Flu, which is influenza. Herpes, HIV or AIDS, and HPV, infectious mononucleosis. Mumps, measles, rubella, you can see here a picture of mumps, and you get there the various other diseases. Now the diseases caused by virus to plants is banana bunchy top, you can see over here, banana bunchy top, curly leaves in chili, and TMV, tobacco mosaic virus. With that, we have come to the end of this lesson, and finally I'd recommend everyone always go through the summary in your textbook. Most of the people tend to either ignore the summary, or they just don't go through it. They think, okay, I already studied everything, I no need to summarize anything. But really this is very important, because if you at least go through, if you read the summary, it will be very helpful, especially in your MCQs. So just read the summary immediately after you finish a lesson, or even before your exams. Rather than going through the entire note, it's good to do some past papers, go through those, and then go through the summaries rather than going through everything, because here it basically has summarized the entire lesson and will give you a basic idea of the whole lesson. So just to go through it, we talked about cellular organization, nutrition, respiration, irritability and coordination, excretion, movement, reproduction, growth and development, which were the characteristics of organisms. And we also said that the basic organizational level of an organism is the cell. Multicellular organisms possess tissues, organs, systems, and level complex organization. The process of obtaining energy and materials to maintain life is called nutrition. Cellular respiration is the process by which end products of digestion combines with oxygen to produce energy inside the cell. Ability to respond to the changes in external or internal environment is known as irritability. The adaptation of the body functions according to those changes is called coordination. Removal of waste products of metabolism during metabolism out of the body is termed as excretion. Organisms move as a result of coordination. Production of new generation for continuation of their species is termed reproduction. Growth is the irreversible increase in dry mass of a cell. During development, cell differentiate to perform a particular function. Virus is an acellular form which cannot be differentiated into living or non-living. All organisms contribute to maintain and for the existence of the environmental balance. With that, we have come to the end of the lesson. So hope you all understood the lesson, enjoyed the video. If you did, don't forget to hit on the like button, subscribe to my channel if you are new, and share my video with others. And make sure you turn on all notifications, because from here on out I will be posting videos that will be very helpful for your O-levels, and in these videos you will be able to get some short notes, and we will be covering up some important parts in these lessons. So do stay tuned. Until next time, bye.